Hello, everybody, and welcome to episode 5 of Astronomy in Context. This episode is all about how humans see color vision and how our eyes operating as a telescope. Now, this is a video that actually spawned the idea for me to create these Astronomy in Context videos, and it's been a bit of a rabbit hole. So everything I'm going to talk about today is just going to be sort of introductory level stuff, and it is not going to go into the depth that it probably really deserves, but this is just such a rich and complicated question. How do we actually talk about light, which we now know is photons that have different wavelengths? And visible light, we can map a wavelength to a certain color. Say hydrogen alpha, wavelength 656.3 is a certain color of red. How do we actually map that wavelength of light coming into our eye, interacting with our retina, and then sending those signals to the brain to actually make us see color? So this is a very interdisciplinary thing where we're talking about how the brain operates. So we're talking about neuroscience, we're talking about astronomy with wavelengths of light. We're talking even a little bit about culture and how culture can affect how we perceive colors, what colors we can discriminate between. So all of this comes together in this really, really interdisciplinary and very, very complicated concept of human color vision. So let's just go ahead and jump right into it and see how we see, or at least how we see color. Okay, so I want to start with what we are contextualizing because we've covered a lot of material since our last video. So a lot of what we're contextualizing here is we've covered telescopes. So I want to talk to you about how our human eye is basically a telescope or a small object that can see an optic. So we have these little telescopes that are equipped in our head, and I want to give you a little information about that and contextualize how our eye is actually operating in the terms that we've learned. And then we're going to get into how we actually see color and how that relates to the electromagnetic spectrum, particularly the visible portion of the spectrum, the colors that humans can see, the part of the electromagnetic spectrum that our eye interacts with and our brain interprets signals from. But let's start with the eye as a telescope. Any good telescope really just needs some lenses or a lens that can focus light and then some sort of a detection element where it can have that image and record that image and have some sort of information so you can sense that image. Our eye is pretty simple. We have what we'd call an aperture, the opening. So you can think about the end of a telescope where the light is going to be filtered through. So it's going to come through the pupil. That would be the diameter. The size of the pupil would be the diameter of our telescope. It'll hit the lens of our eye, which will then focus the light. And it'll put the image of what we're seeing here depicted as a tree. It'll hit our retina. And then we have basically photoreceptors in our retina, which are going to be specialized cells that can detect light. So they have proteins that are light sensitive. And then that gets sent by our optic nerve to our brain, and we can see an image. Notice that our eye actually flips the image of the tree, and then our brain reinverts it so things look upright and we don't see the world as upside down. There's actually been an interesting experiment done where people have invented glasses that invert the world, and people will voluntarily wear them for a while. And they experience extreme nausea, fall down a lot, don't feel very well for the first while. But then, after several hours of wearing glasses that literally flip the world upside down, the brain will self correct itself and reinvert the world so everything looks normal and you can operate as a normal human again, which is kind of crazy to think about. And then you have to go through the entire sort of reinversion process when you take those glasses off. Something that if I had a bunch of free time and I felt like I could deal with feeling sick for a while, I'd really like to experience that. Okay, so that is how our eye is seeing. But let's relate this to some of the things that we've talked about. Angular resolution is how well something can see, how much detail it can see. Can you see two individual points and discriminate them as individual points of light, or are they blurred together into some blob and you can't see any detail? So, what about the human eye? How well can the human eye see? So, what is the angular resolution of the human eye? How close can two objects appear next to one another? You can see them as distinct objects. And for this, it's really important to talk about the angular resolution of the human eye in daytime, bright light, and nighttime, because as the environment gets darker, our pupil will dilate, it'll become larger. So we have an adaptive diameter of our face telescopes here. So the angular resolution during bright light, our pupil is about 3 millimeters on average for humans. It varies from human to human and how bright things are, but it's about 3 millimeters. is the diameter of our face telescope. That's about 0.003 meters. I could plug that into our angular resolution formula, where angular resolution will be in arc seconds. And then the wavelength of light is going to be in microns, and the size of the telescope, our face telescope here, is going to be in meters. And go ahead and crunch all those numbers. So it's 0.25. So the middle of the visible spectrum is about 500 nanometers, zero point 0.5 microns. So put 0.5 divided by the diameter of the telescope, 0.003 meters. And the angular resolution of the eye during the daytime is about 42 arc seconds. Just a little bit less than an arc minute, a little bit less than a 60th of a degree. That's pretty darn good. But at night, whenever it gets darker outside in your darker conditions, your pupil will dilate and it'll get to be about double the size, so about 6 millimeters, which means that your angular resolution actually goes up. The problem is that there's less light, so you can't really see things as well, and you really can't see color. You switch to using your rods instead of your cones. So it's a lot harder to see at night, but technically you have better angular resolution because your eye pupil is bigger. And in fact, notice that it's getting about double the size, so you end up with half the angular resolution of about 21 arc seconds, a little bit less than half an arc minute angular resolution. Okay, so now we see how the eye can see. We're taking light, we're focusing it with the lens of our eye, and it's falling on our retina. That doesn't explain color vision, though. What's going on with that? And for this, let's go ahead and review the electromagnetic spectrum so we can put this idea into context so you can appreciate what's going on. So the electromagnetic spectrum of light, or really just the electromagnetic spectrum, is all the possible wavelengths of light, all the possible colors of light. So you have very, very long wavelengths, radio waves, and then you have very, very, very short wavelengths of light, gamma ray light. And then right here, this very narrow region in the middle is the visible light spectrum. So this is what the human eye can see. So we see, and oops, I didn't mean to say we see a very small portion of the electromagnetic spectrum. Very, very narrow range, but this is what the human eye can see. So we can give it colors like red to purple. But how does this actually translate to color? Because what is actually happening here? We just have an electromagnetic wave that has a different wavelength. We just have a photon that has a different wavelength, a photon that has a different frequency, a photon that has a different energy. If you're thinking about the particle of light, the singular photon. And these things are coming from all the cosmic sources, coming and reflecting off stuff. Some of the lights, some of the wavelengths are getting absorbed, some are getting reflected. Sometimes you're just looking at that light. If you're looking at the stars or the sky, that light's just coming right into your eye. It all has different wavelengths, and it's going to enter our eye through our pupil, get focused onto our retina, and somehow we're going to see color. How can we actually say, hey, that photon was a green photon, that photon was a blue photon? How do we see color? It's just an electromagnetic wave with a different wavelength. That is complicated but super duper cool. So how do humans see color? Does our trichromatic three-color vision come from? And for this, we have to look at the details of what's going on with our eye, and particularly what's going on in our retina. So we're focusing the light is going to fall on the retina, and clustered right around this area right here, we have a lot of specialized cells called cones. The retina itself is full of both rods and cones, which are specialized photoreceptor cells, which photoreceptors means that whenever a photon hits it, it reacts. For our eye, this is an electrochemical reaction. So there's going to be some sort of electrochemical that signal. So there'll be an electric thing that gets sent along the optic nerve that then we use chemical transmitters, neurotransmitters, that signal will get interpreted to the brain. Kind of crazy in its own right. I told you this is a rabbit hole of a video. But we basically have rods and cones which can sense light. Our rods see black and white. So our rods just tell you how bright something is. So they can just say, hey, you see it or you don't. And the photoreceptor there is this thing called rhodopsin. Cones, however, are much fewer. You have about 1,000 times more rods than you do cones. The cones have photoreceptor opsin. Sorry, I didn't mean to put an extra I there. They have opsin proteins that use a certain thing called 11-cis retinol and its variations as their chromophore, the things that can sense light. And we're equipped with three different kinds of cones, or at least humans are. It varies from animal to animal. But humans have three color sensing cones. So we have cones that are specialized to detect red photons. We have uh, cones that are specialized to detect green photons and cones that are specialized to detect blue photons. We have a lot more red cones than we do green cones. We have a lot more green cones than we do blue cones. But these are our photoreceptors. And let's take a look at actually what's happening here. So we have these three different cones, our red, green, and blue cones, which are called the L, M, and S codes. L for long wavelengths, M for medium wavelengths, S for short wavelengths. And they can detect photons over a range of wavelengths. And that's what this is showing right over here. So here we have the wavelength on the x-axis, and then how sensitive it is to detecting that photon. So how many of those photons, if you send out 100 photons, how many of those photons would actually be detected? So you can see if we look at the sensitivity of our blue cone range, it goes from about 400 to 500 nanometers. And it has a peak sensitivity somewhere between 420 to 440 nanometers. And then we can go to the green, and it ranges from about 450 all the way to about 630. So if you have a photon of that, the green cone will be able to sense it. And it has this peak at about 534 to 555 nanometers. So sensitive to, let's see if I can actually give you a green blast with a green laser pointer. So here is green. This is 532 nanometers. You can see right there. Do I dare? Yeah, so you can see the green right there. That's 532 nanometers. So right about here. And then we have the red, which is sensitive from about 500 to 700. So any photon that has a wavelength 500 to 700 nanometers can react with the red cone, and your brain can sense that red color, which is kind of interesting because that tells us that humans really only see three colors. We see red, green, and blue, RGB. And what red, green, and blue actually are defined by the sensitivities of our cone. So that is actually what defines RGB, red, blue, and green as colors. Crazy, right? So now we see that this light is coming in from all the sources around us. Take a look around you. You can see things. I see some green on my plant over there. I see some blue in the sky. I see some blue on my globe back there. And all that's happening is the light from there, you're getting a range of wavelengths that are entering my eye. And some of them are within the blue range, some are in the green range, some are in the red range. They pass into my eye, they hit my retina, where then these cones can sense them. And they can sense basically how many of each of these photons are coming in. And I'm going to come back to that facet right there. So we can sense red, green, and blue, and how many of each within that wavelength range are hitting our eyes. And it's going to be that balance that actually lets us see color. But let's hold on to that because that's where things get interesting. I want to go ahead and throw in a little bit more context right here. You might be wondering, if the electromagnetic spectrum is so broad, there are so many different parts of it, why do we see visible light? And this is a natural consequence of evolution. It's no surprise. If you look at the solar spectrum shown here on the left, so here's the light from the sun, and the back in the bright yellow, you have a black body at the temperature of around the sun. And then you have what hits the top of the atmosphere, where, well, where you have a little bit of the atmosphere above it doing some absorption. And then this is what reaches the ground. So you can see most of the light that reaches the ground is visible light. So it's not a surprise that that creatures evolved to react to the most abundant light source. And just to kind of overlap that, so we could go ahead and take a look at all of these sensitivities. So here we have our red, or here we have our blue, our green, and our red. The black right here is the black and white from our rods. So we have all of those, but let's just pay attention to the blue, the green, and the red, and put them all together. So that would be called a convolution to see how sensitive we are to each individual wavelength of light. So what wavelength of light are we most sensitive to? What color is easiest to see? That's what we have over here. This is the sensitivity, and we're most sensitive to green light at about 550 nanometers, which is exactly where the solar spectrum is peaking. Hmm. So, no surprise at all that we humans specialize in seeing visible light since it's such an abundant resource from our sun. So now you can start tying all these things. We got some biology going on, we got some astronomy going on, we got some psychology and neuroscience going on. This is just such a cool subject in of itself. But now I told you to put a pin in the fact that we only see with our S, M, and L cones, our blue, green, and red cones, and we can only detect they either got hit by a photon that has a wavelength somewhere in this range, or they didn't get hit by a photon. So all of these photons are entering our eye and getting absorbed by our rod so we can see black and white, and our cones so we can see color. And what is actually happening as these come in? Well, we're detecting how much red we see, how much green we see, and how much blue we see with our cones. So we have this balance of how much red there is, how much blue there is, and how much green there is. That's all we can tell. Like if we send in a yellowish photon, it's going a whole bunch of yellow photons. Some of them are going to get absorbed by the red cones, some are going to get absorbed by the green cones. So we'll have both green and red going, hey, I see those. So we see green and red simultaneously. If we send in a yellow photon, we get activation of both of those cones. If we send in, say, a uh, shorter wavelength green photon, sort of a cyanish photon, then we're going to get a little bit of red, we're going to get a little bit of green, and a little bit of blue activation. So we have all of these coming in, and we get some balance of, hey, we're detecting this number of green photons, this number of blue photons, this number of red photons. We can put that balance of how many of each photon we're seeing into what's called a tristimulus value. It's a very fancy term, but it's basically saying, I saw this much red, I saw this much green, I saw this much blue in this ratio. That's what our eye physically sees. Then they get sent to our brain via the optic nerve, and our brain interprets that tristimulus value, that balance of red, green, and blue cone activation goes, well, what you're seeing is yellow. What you're seeing is blue. What you're seeing is some other color. So we have a combination of how much R, how much G, and how much B gives us all the colors that humans can see. Crazy, right? Absolutely bonkers and super duper cool. And now we can play around with, and I think this is one of my favorite little facets that comes to this is, all right, let's take the color pink. What the heck is pink? Because if we can only physically see red, green, and blue, that means our brain and our interpretation of the balance of red, green, and blue is just making up all the other colors. We are just perceiving all the other colors as a balance of those three R, G, and B detections. So let's take the color pink. The better way to describe pink is not green. So here I have in the square, red on this side, blue on this side. Nary a green to be seen. I cranked up the red to 100% red, cranked up the blue to 100% blue with no other things. And then I just blended them together. And you can see where they're blending. You see pink, where you're getting an activation. Your red cones are going, hey, I'm detecting red, I'm detecting red, I'm detecting red. Your blue cones are like, I'm seeing blue, I'm seeing blue, I'm seeing blue. Your green cones are like, they're probably a little bit activated since there's so much overlap between red and green. But for the most part, like, I'm not getting much green. So you have a whole bunch of red, you have a whole bunch of blue light coming into your eyes, and your brain goes, that's pink. I see pink. That's definitely pink. And I want to go ahead and give you a physical demonstration that pink is not green. So here I just have a white circle, and give me a second here. So what I'm going to do is I'm going to physically manipulate the color of this circle. So let's click on the circle and let's change its color. All right, so now I have full control over my colors. I'm going to go to my red, green, and blue sliders. And all I'm going to do is I'm going to remove all the green. No green. What color do I get? Pink. Or technically this would be magenta, but you can say pink and magenta are relatively synonymous. It's a little bit different. If you want to go into like color schemes and color theory, you got to do a little bit of nuance there. But if I were to ask you, hey, what color that is, you'd probably say pink instead of like, ooh, that's definitely magenta. I'm not going to go to the additive and subtractive color models here. If you're interested in those sorts of things, by all means, use this video as a launching point. So many cool things. Now we're bringing in the whole art world and color theory. And if I was an artist, I'd definitely get sucked down the rabbit hole of pigments and developing different colors. Like the fact that they just discovered a new blue and they're selling a new blue that humans have never seen before, I kind of want to go buy that on the internet. All right, so now let's go and take this idea and run with it and talk about the colors of stars. Because if you look out, and this is just looking at a globular cluster, cluster of a whole bunch of stars, hundreds of thousands of them, some millions of them, and all in the same region of space, tightly packed together. So this globular cluster of stars, you take a look at it, you see that some stars are red stars, some are orangish stars, some are blue stars, some are whitish, and these are the colors of stars that you see. These are our spectral classes of stars, which are basically just how we categorize stars based on what their spectra look like. And then we can map that to color, which is dictated by their black body part of their spectrum. But these are the colors that we can see. We can see blue stars, blue white stars, yellow white stars, yellow stars, light orange, and orangish red sort of stars. These are all the stars we see, which is really kind of interesting. Because why don't we see things like pink stars? Why don't we see purple stars? We know by Wien's law that the hotter you get a star, the more shorter wavelengths it'll give off. And if you go to really, really hot, you'll have the peak of the black body spectrum in the ultraviolet, and it'll be giving off a whole bunch of violet light compared to how much red light is giving off. So why don't we see purple stars? This is where we have to go into the fact that we have red, green, and blue, and all those colors are coming in. All those photons are coming in. So when we look at the stars, we're getting violet light, blue light, and then green, yellow, orange, and red. All of that, we have to think about the balance of all of those, how our cones sense those, what signal it sends to the brain, and what our brain interprets that color as. And we have to ask ourselves, will our brain, based on the black body spectrum, the temperatures of the stars, ever actually interpret and see the color purple? The answer is no. And now we have to go into another cool part of how color works. This here is called chromaticity. And right here, we're looking at a color space chromaticity diagram. And let me just go ahead and break this down a little bit. X roughly correlates to various values of red, blue, and green happening, a mix of all of them together. Y axis here is mostly blue to green, bluish, bluish axis, but it combines a little bit of RGB and all of that. It's a really complicated thing, and I'm not going to go into how you get all of them. I got lost for days down that rabbit hole, but I can describe what's going on with the rest of this without us really knowing exactly what an X value or a Y value means. Around the outside, we have what are called a spectral focus, which is, notice that this goes from 380 nanometers of light. So this is if you just had a laser beam at 380 nanometers, you shined it, that's the color it would be. And then you can go from 380. And if you go around the outside of this, you're getting all the spectral colors, which is just the rainbow. And then as you go into the middle, you're getting different balances of red cone, blue cone, and green cone activation, which gives you all the possible colors that humans can see. What this leaves off is brightness. So you can change the brightness. This is just the different colors. And it's all the colors that the human eye can see. Now let's take a look at black body spectra of different stars. So I have a star at 25,000 Kelvin, 6,000 Kelvin, and 3,000 Kelvin. And here's their black body spectra as it goes through the visible part, which I've put a purple line to a red line, violet to red. So you can see in this 2500, we see a whole lot of violet colors and a little bit of red, which means all those photons from that starlight are entering our eye, and we're getting our blue ones going, holy crap, that's a lot of blue. We're getting our green ones going, that's a lot of green. And then we're getting the red ones like, I don't see much red, but it's there, I'm still detecting it. And then our eye is going to blend all that together. And we're going to end up seeing this as a blue star. Why not a purple star? Let's come back to that in a second. Let's go to 6,000. Here's the black body spectrum through the visible range. Now let's consider what we see with, say, the blue. So we see a good bit of blue. What do we see with our green cones? Oh, we see a good bit of green. And what do we see with our red cones? We see a good bit of red. We see about all of them equal in equal parts. So our blue, green, and red cones are like, yeah, I'm seeing about all of those wavelengths of light in equal measure. What happens if you combine all the colors of the visible spectrum? Well, you see white light. So a star like our sun, if you can get out of our atmosphere and look at it, our star would look white. It's a white star, maybe a little yellowish white. And then let's go to a really cool star, a star like Betelgeuse. So if you go and you find Orion, it's the shoulder of Orion. It's a really, really very apparently red star. It's a red supergiant star. And it has a surface temperature of about 3,000 Kelvin. Here's what its black body spectrum would look like through the visible range. So you don't get very much blue. You get a good bit of green going on, and you get a whole lot of red going on. When the red heavily outweighs, so what do you see? You see mostly red, and your eye or your brain interprets those R, G, and B values from your cones as reddish. And you could imagine I could do all the temperatures between, and I could figure out how our eye would respond to that balance between our red cone activation, our green cone activation, our blue cone activation. And I could translate that to what color our brain would interpret that as. And that's what this line is right here. So these numbers, this is a temperature, a black body color temperature, as we'd call it, of 1500 Kelvin, 2000 Kelvin, 2500, all the way up to if it were infinitely hot, which doesn't really exist. So just think of it as unimaginably hot. So these are the colors that we can see from black bodies, black body radiators. And notice that, yeah, the cooler ones appear red, and then they appear orange, and then they appear sort of yellow whitish, and then whitish, and then they appear blue. We have sort of blue whitish, and then we have blue, but never purple. So there are no purple stars. There's no way, since stars act as a black body, that you can get a spectrum of a star that would give us blue, green, and red cone activation so our brain senses purple. Just can't have it. So there are no purple stars. Whew. All right, so we've covered a lot of territory there. Slight little break, because now you're thinking, I have cameras and my phone. How do they see color? And why do astronomers always have their most detailed pictures in black and white? Surely NASA is using the most advanced camera technology. Why won't they have color cameras up there? And the reason comes down to, how do we actually make color images with a photograph? So this is a little bit of a twist and turn to not how humans see color, but it's based off how humans see colors, how we design cameras that can create color images. Because if we can only detect red, green, and blue, and if we have something like a CCD or a CMOS detector, so if you pick up your phone, it almost certainly has a CMOS detector in it. Older phones will have CCD detectors. And all these is basically, if you were to zoom in on it, so here we have a very zoomed in image, you'd see that you have these little rectangular buckets that are photoreceptors. They're light sensitive. So a photon comes in, will hit one of those pixels, and in that pixel, that photon will release an electron. It will knock free an electron out of the material that the pixel is made out of, that the CCD chip or CMOS chip is made out of. This is called the photoelectric effect. Thank you, Einstein. And then basically, so you'd have a photon come in, visible light photon has enough energy, knocks free an electron, and then you count up the number of electrons, and that's how you make a image with a CCD or a CMOS camera. However, the photoelectric effect, it has a threshold for, oh, you need to have a wavelength of this certain value, this much energy in a photon before you'll knock an electron free. But it can't tell you if you're above that threshold. It can't go, well, that was a green photon, or that was a blue photon, or that was a red photon. It just says, yeah, I saw a visible photon. Could have been an ultraviolet photon too. So I got a detection. Great. And then you read off how many detections it got, and you have an image. But that's naturally black and white. So how do we make color images? Why can you just pick up your phone and take a picture and it's a color image? And there are several ways of doing this, but the most common way and the cheapest and easiest way is to put little tiny filters. So if you were to take out the chip that actually records the image on your phone and you were to look at the detector, you'd notice that some of them have a little green filter over them. Some of them have a blue filter over them. Some of them have a red filter over them. So the filter makes it so only red light will come through, only green light will come through, only blue light will come through. So each pixel is recording a R, G, or B value. And then you can put that together. If you show that back to a person, then and you color all the red ones red, all the blue pixels blue, all the green pixels green, based on how bright their green, blue, and red brightness was. You put filters, so now you're actually sorting the photons, similar to how our eye does with its cones. Then all of a sudden you've recorded a color image because your brain will see that and turn it into a color image by blending the green, red, and blue values, just like we were talking about with how our cones respond. So that's how you make a color image. And the most common pattern that people use is called the Bayer pattern. And you can see how they'll have this is the pixels that are mapped to, that have little red filters. These are the ones that have blue filters, and all the rest of them are green filters. So that's how a CCD camera, you can take a color picture. But notice now you're throwing away a lot of photons because you're getting rid of the green and blue photons here, the red and blue photons and the red and green photons. In astronomy, we're trying to see the faintest things out there. We don't want to throw away photons. So we don't use this in astronomy detectors. And in fact, if you want the most detailed, the highest pixel resolution images, you're not going to look at a black and white. If you want a color image in astronomy, what you do is you don't build a pattern on your chip like this and throw away a bunch of photons. What you do is you use color filters, like a blue filter, a red filter, and then a green filter. These are official astronomy filters called the UBVRI filters: ultraviolet, blue, visible, red, and infrared. These are the response, how sensitive they are to wavelength. So here we have wavelength and how sensitive they are. So really, whenever you use these astronomy filters, you're using 5 color vision instead of 3 color vision because you're adding in ultraviolet and infrared. But then you have blue, visible, and red. And you would take a picture with the red filter in place, take a picture with the blue filter in place, take a third picture with the green filter in place, stack all 3 of those on top of one another, got maximum use out of your photons, although you had to take 3 images, stack them all together, color the red ones red, the red image red, color the green image green, color the blue image blue, and you get a color image. Which is why it usually takes a while. If you're seeing a space mission, they'll send back black and white images before they'll send back color images. Because a lot of times they'll have the camera that has these filter wheels in there where it can take green, blue, and red pictures. But then you'd have to send back 3 times the amount of data. So just a little bit of bonus extra stuff. And now, if you can still hang on, I want to play with something kind of crazy, some optical illusions, or what's really called impossible colors, colors that you can't physically see unless you wear out your cones. You oversensitize, you give too much stimulation to some of your cones, And then in an after image, the human brain, since you overstimulated some cones, you'll end up with an after image of a different color that you can't normally see, so-called impossible colors. So maybe you have seen this. The way I'm going to do this, you can pause and rewind. You have to choose pretty much one at a time. You can kind of do all three at a time, but just pick one of these three circles. And what you're going to do is you're going to stare at the X. And eventually I'm going to take away the colored circles. So this yellow circle, this green circle, or this cyan circle. And you're going to see a after image that's roughly circular in shape of a certain color. As we wear out the cones that get used to see this yellow, we're going to see a residual blue color that's called Stygian blue that you can't normally see. If you stare at this green one, you can do it and you can see a color called self-luminous red. If you stare at this blue one, you'll see a color called hyperbolic orange. And this is happening because we're fatiguing our cones that detect yellow. So whenever that stimulus is taken away, our brain actually gets confused and sees blue as an after image. And in this case, it would be a color that we can't normally see, because if you notice, I go ahead and go back really quickly. If we go back really quickly, notice that there's no way you can just see green. You send in any any photon that would be detected by our M cone, our green cone. It'd also be detected by either red or blue, so you can't just see green. So this color hypergreen doesn't really exist. Although you can perceive it if you wear out your your red cones and your blue cones, you'll get an after image that is a green that you can't normally see. All right, so let's actually play this game. Pick yours and stare at it. And then in about 10 seconds, stare at the X. Don't look at the other one. Stare at the X. And in about 10 seconds from now, I'm going to take away the circles and you'll see the after image. I'm looking at Stygian blue right now. So you should see after images, and they each have their own colors. So I encourage you to go and do that as much as you want. If you want to see hyperbolic green and a cool optical illusion, let's take a look at what's going on with this weird slide that I just showed you. And now we're in sort of the fun part and bonus material of the video. Notice that we have a bunch of magenta circles. And basically one of them is not there and it rotates around and around and around. So what I'd like you to do, this is activating your red cones and your blue cones and not your green cones. Remember, pink is not green. So stare at this black dot and you'll see this green circle, this hyperbolic green, a color that you can't normally see, is replacing what was a blank dot, where the blank dot was. So you see a green dot, the color green that you can't naturally see, moving around and around and around. And for me, the more I stare at it, sometimes the magenta circles just start disappearing. Oh, I lost all the magenta circles. Now I see some of them. So I'll leave this sitting up for a minute for you to enjoy. You can also just look it up if you would like. I'll try to put a link into the video or next to the video whenever I drop this video. I hope you enjoyed how the human eye sees color, how we build color images, how that relates to the electromagnetic spectrum, and how we have these little face telescopes that are equipped with super cool detectors, biological detectors that take electrochemical signals and translate them into the colors that we see. So we're going from an electromagnetic wave. Oh yeah, that's the bonkers part. So here we just have these little bundles of coupled electric and magnetic fields, photons, an electromagnetic wave hurtling through space, created by things that have a temperature, going throughout space. They're going to enter our eye, interact with these specialized cells that we have in our face telescopes, and then our brain is going to be able to detect certain balances of how many of each one of those photons we saw, each one of those little packets of electromagnetic waves, and all of a sudden we have a beautiful color world that we see. Crazy, crazy cool stuff. I hope you enjoyed this astronomy in context. I will see you in the next one.